This is your brain. About the size of a grapefruit, it's a squishy, fatty blob that contains everything that makes you, you. Your hopes and dreams, your talents, all the things you've learned, and all of your embarrassing memories. It also takes care of a lot of things you don't normally have to do consciously, like breathing or finding a comfortable place in your mouth for your tongue. Both of which you're now having to do manually just because I reminded you of them. You're welcome. Brains are some of the most powerful computers we know of, but brains have a problem. Well, two problems. The first is that most of the compute power of a brain is spent on guiding behavior that keeps the brain alive. After all, every animal has to navigate in 3 dimensions and contend with a wide variety of environmental factors, all while trying not to get eaten, find food, and find a mate to reproduce with. The second problem is, of course, the lack of convenient auxiliary ports.
This will feel a little weird.
But what would happen if we took some neurons out of a brain and grew them separately? If we give them nutrients to keep them alive and a new set of signals via some electrodes, can they learn to do other things? And if so, is there a limit to what tasks they can do if we choose the signals correctly? Let's find out. Inside this weird-looking container is approximately 1/200th of a rat's brain, or approximately 75,000 living neurons. This container is our latest prototype of what is called a multi-electrode array, which is exactly what it sounds like. It's a special arrangement of electrodes, embedded in plastic for the neurons to grow on, so that way we can pick up signals they emit and input signals for them to process. So what signals are we putting in? Well, other groups have used neurons like this to navigate around in mazes, control small robots, or even fly planes in a simulation. But we have more ambitious goals. We want our arrays to play the classic video game, Doom! Depending on how you're counting, this video is actually the 3rd part in an ongoing series, where we're working towards building a system capable of playing Doom with living neurons. We're calling it the Torment Nexus, named after the fictional sci-fi book, Don't Build the Torment Nexus. And thus far, we've been making great progress. Let's have a look at our tech tree so we can see where we are. And there have been some unexpected side quests we need to add on here. First, we had to build a special incubator to keep the cells alive, and we tested that by growing skin cells, which are cheaper and much more forgiving than neurons. We also spent several videos building a plasma-based metal coating system to try and make our first arrays. Our first attempts at growing neurons were pretty disastrous, so in the last video we focused solely on that aspect so we could perfect it. And we also tested some alternative methods of making the arrays that were easier than the plasma coating method. Recently, you also saw our side quest of growing cells in Gatorade and other sports drinks. That was actually a pretext to start developing our own cheap growth media to cut the cost of these experiments, from hundreds of dollars to tens of dollars. And while our recipe still isn't perfect, our first attempts to cut the cost of growth media worked really well, all by using a Japanese sports drink. And finally, here is a side quest you didn't know about. Part of the control scheme that we have in mind requires us to not only have a living array of neurons, but a simulated partner. That way, we can test stimulus on the simulated array before sending the command to the real one and try and get an idea of what's about to happen before it does. I won't get into the details now, but it does mean that we need to get comfortable simulating neurons. Long term, I don't want to just simulate a handful of neurons though. I want to eventually build up to simulating a whole brain. So anyway, here is the first step towards all of that. What you're looking at is 1 second worth of simulation of a single cortical microcolumn. These are the building blocks of a brain, and in this visualization, you're only actually seeing a tiny fraction of the true number of connections between the neurons. Here's what 1% of them being visualized looks like for context. Eventually, we'll use what we learned from these simulations to improve the connections between the virtual and real neurons. Today, though, we're focused on the physical side and taking the next step towards playing a game. We're going to be focusing on actually getting signals out of the neurons and getting used to stimulating them in various ways to see how they respond. And we're going to do this both electrically, but also visually with the help of an amazing die that we're going to talk about shortly. Now, in order to do this, we needed some very serious hardware. First, we needed better arrays than last time because our homemade attempts haven't been particularly successful. Ideally, we needed commercially made neuron arrays so that at least we had something that we knew for sure would work. Then we could study them and see if we could improve our homemade ones enough to get those ones working. And if we've got commercial arrays, it would be amazing to also have a commercially made recording system so that way we can get some data on what the signals really look like as the neurons grow, so we have a real baseline. Well, wouldn't you know, after the last video we got in touch with the wonderful people from Multichannel Systems, and they offered to help us out on both fronts. They very generously sent us a bunch of their amazing neuron arrays to use and lent us one of their MEA 2100 mini systems so we can take our first real recordings. We'll talk about just how cool this amazing machine is in a bit, but huge thanks to them. This alone has pushed the project forward practically light years. Let's start by looking at some of these arrays because they send us a variety of the kinds that they carry, and frankly each is prettier than the last. All of these arrays were made using plasma deposition, and they're all made of glass. These arrays either have electrodes made out of titanium nitride, or if you want to see every little detail, they also carry electrodes made of indium tin oxide. In either case, the electrodes are only a few micrometers wide. We had pretty much given up making arrays like this because of how hard it was, so I'm pretty excited to try these out. One thing that has been a huge problem for us in the past was how we seal our arrays. These cultures are very sensitive to contamination, so they need a lid, but they're also living organisms and need to breathe. The latter part is actually the harder of the two to contend with, because most of the things that give a good seal also prevent air from getting in, so our cultures kept suffocating. These commercial arrays use a very clever system of a Teflon ring with a thin sheet of FEP film stretched across it, which is held on with an O-ring. FEP film is the same stuff used in 3D resin printers. It's a fluorinated plastic that prevents water from diffusing through it, but has pores small enough to let oxygen and CO2 pass through relatively easily. And FEP film is super cheap. I picked up a roll of the 25 micron thick film for all of $25. Now, before we get into the weeds about our new batch of homemade arrays and our new control scheme for getting the neurons to play a game, I want to show you how all our new tools work. For that, we need some neurons. As usual, we'll be using these prepared tubes we buy from the manufacturer that contain about 1 million neurons ready to go. They're massively overpriced for what they really are, so we're working on a cheaper alternative, but for today, they'll work well. We went through the process of how to culture neurons in detail like this in the last video, but after we posted it, we were contacted by Dr. Steve Potter, who is a legend in the world of neuroscience. He had an entire lab dedicated specifically to using cultures of neurons to interact with a computer, and had a ton of experience using these exact arrays. So on his advice, we made some changes to the protocol. To get the neurons to stick to either the arrays or just the bottom of a normal 12-well plate, normally you have to apply a chemical coating. Last time we used something called poly-D-lysine, which kind of worked, but at higher densities the neurons would sort of peel off and start to form these weird brain orbs. This time we're using a double coating, on Dr. Potter's suggestion, of polyethylene imine, also known as PEI, followed by a layer of laminin. PEI is a synthetic polymer, while laminin is a protein mixture. To apply the coatings, we put a solution of PEI into each well or array and then let it sit for half an hour. Then we draw it back out, rinse everything well with sterile water, and let it dry under the clean air of our flow hood. Then, when it's dry, we apply the laminin and then let everything sit in the incubator for about 20 minutes. After that, we can remove the excess laminin and add our culture media. Again, this is where we're testing out some different things. Dr. Potter pointed us to some literature that showed that, contrary to what is commonly done, ordinary, cheaper DMEM gave electrophysiology that was more similar to what you get in an actual brain. He suggested that neurobasal media, the stuff that we used last time, was optimized for cells looking nice in pictures rather than electrophysiology. Well, we've got a fancy recording system and a bunch of cool dyes, so we can just test that and find out which we prefer. So we set up one array with neurobasal and one array with DMEM, supplemented with horse serum and insulin. Why those ingredients? I have no idea, but there'll be a pile of links in the description to papers for those interested in the exact recipe. We also set up 8 wells of a 12-well plate with either DMEM or neurobasal so we can have a look using our fancy new dyes and see if we can visually tell the difference in their activity. After everything had time to warm up and equalize in the meat cubator, we could add our neurons. Now, these will need a few days to cook before we can really do anything with them. It'll take about a week and a half to 2 weeks for them to really establish the network and be really usable. So it's pretty important that nothing happens to them in that time. Otherwise, you'd have to start over. Hey, how are the neurons growing?
They all died.
Fuck! Yeah, I don't know what happened, but the first batch died. We have no idea why. But thankfully, these arrays are reusable. To do that, they get a long soak with an enzymatic solution of trypsin, followed by a thorough rinse to clean out any of the dead cells. They can then be re-sterilized by dunking them in 70% alcohol and allowing them to dry in the hood. Anyway, skip to a few weeks later when a new tube of cells had arrived and had time to grow. Here they are a week into the culture period. As you can see, there's a dense network already forming. But let's take a peek at what they're thinking about and try out our new dye. Today we're going to be using a really cool one called CalBrite 520 AM, which I'm just going to call CalBrite. What makes this dye amazing is it has two special properties. The first is that in its base form, it doesn't actually look like anything. No fluorescence, no color, just a clear solution. But when it gets inside cells, enzymes in the cells react with the dye to convert it into a fluorescent form, making it way easier to see the cells. And only live cells should glow. The second property, though, is that the fluorescence isn't uniform. In fact, the brightness of the glow should be proportional to the level of calcium that is present. Now, you might be thinking, well, why does that matter? Well, when neurons fire, a couple of different things happen. The first is a rush of sodium and potassium ions into and out of the cell. That's where the electrical current comes from when a neuron fires. But that rush of moving ions also triggers the influx of a huge amount of calcium into the cell. So cells stained with CalBrite should glow much brighter right after they fire. These cultures already show lots of spontaneous activity where random cells will pulse, though we did notice that the dye seems to saturate the longer you look at it. If you look at any one area for too long, all the cells eventually are just in the bright state and you can't see the pulses anymore. But I think this is just a quirk of this specific dye. It must need a while to reset to the off state, or something about the illumination makes it get stuck in the on state. There are alternatives to this dye that can be encoded genetically, and we've actually been testing those in the background. For example, a specially engineered protein called GCAMP6F can be delivered to cells as a piece of DNA, and has similar properties to CalBrite. However, its reset time is way better, and it doesn't get stuck in the on state as much. And since it's delivered as DNA, you can also choose the exact cell type you want to express the protein. For example, muscle cells also use calcium flux as part of their firing mechanism, so GCAMP expressing muscles light up when they fire. But we're not here for muscles, we're here for brains. Several years ago, a group of researchers produced a strain of zebrafish that were transparent enough that you could actually see through their skull, and then used GCAMP specifically expressed only in their neurons. The result is this amazing footage of a fish that is thinking as it's swimming around. You can literally see where the abstract thoughts are versus where the navigational and muscle control bits are. Part of the reason I even found CalBrite was because I was looking for ways to replicate this experiment. The idea of being able to see the electrical activity was just so fascinating to me. And when I found CalBrite as a cheaper and theoretically easier alternative, I thought I'd give it a try. But now having tested it, I can see why GCAMP is preferred for a lot of applications, even if genetically modifying neurons is harder than adding a drop of dye. One last thing to try with the dyes is chemical stimulation. We tried a few different things, but by far the most interesting was capsaicin, also known as the spicy molecule. As you might expect, adding some capsaicin to the cultures causes the neurons to fire more. They also start releasing bursts occasionally, where a huge amount of neurons light up all at once. But to dial up the heat, we just dropped a few flecks of cayenne pepper right into some of the wells. You can literally see the wave of capsaicin pass through the liquid. As the wave spreads, the neurons all kind of get stuck in the maxed out state. But I don't know if this means they're not firing, it could just be an interaction with the dye. Okay, we're comfortable with the dyes, so let's look at the new recording system and take it for a test drive. Now, theoretically, this amazing system can not only do live recordings, but it also has a hole in the bottom for a microscope objective. So in theory, we could look at the neurons firing and see the electrical activity at the same time. But when we tried this, we couldn't actually zoom in close enough to see the blinking effect, and you only see it when you're zoomed right in. At this magnification, you just see the flat glow. But that's okay, because the readout from the software suite that comes with the system is amazing. Not only do you get to see the electrical activity of every electrode individually, but there are all sorts of tools built in, like filters, spike detectors, and analysis tools to make any experiment a breeze. The machine itself has 3 parts. The head stage, where you put the array, a signal collector unit that can handle up to 4 head stages at once, and the interface board that the collector unit plugs into. This is connected to a computer with a USB cable, though make sure you've got a big hard drive installed because this thing churns out data like it's a full-time job. You know what else churns out data like that? You. Without realizing it, every time you use the internet, you're leaving a trail of crumbs that data brokers hoover up, package, and sell to the lowest bidder. Search histories, names, addresses, and so much more, all available to whoever wants it. Between data breaches of your favorite websites, honest mistakes, and unknown vulnerabilities, there is a lot of your data out there. And while you might not think that matters, those with nefarious intent would disagree. For example, if you're in the hellscape that is the United States, your ability to get medical treatment is controlled not by your doctor, but by health insurance providers. And they are always looking for any excuse to raise your rates, and are big clients of data brokers, as are the scammers, fraudsters, and other ne'er-do-wells looking to make a buck at your expense. Enter the sponsor of this video, Incogni. Incogni takes the fight to the data brokers and uses an endless barrage of removal requests and other tools to force them to remove your data from their collection. And they continuously monitor in case your data pops up again, so they can immediately start working to get it removed. If you use the code ThoughtEmporium, or go to incogni.com/ThoughtEmporium, you can get protected now for the incredibly low price of about $62 per year if you go with the annual plan. That's just over $5 per month to not only get the peace of mind knowing that your data is safe, but also the knowledge that you'll stay that way. So head to incogni.com/ThoughtEmporium or click the link in the description after this video and get protected today. Now, back to the machine whose data you all came here to see. To load an array, you just press the release button to open the clamshell. Just drop the array in carefully and close it up. Then you just fire up the control software. There is an included calibration tester that makes simulated neuron spikes and other signals that comes with the machine we're supposed to use first. With the calibrator installed, when we turn on the DAC, we can see the rhythmic pulses it puts out onto every channel. This means that everything is working as intended. However, when we switched over to an actual array, we found that there was a huge amount of electrical noise leaking in, making the signals really hard to see. So we made ourselves a little science neuron burrito and wrapped the head stage tightly with aluminum foil. The case of the head stage is grounded, so this turns the whole thing into a Faraday cage, and makes taking readings much easier. Though we're going to build a better Faraday cage for future experiments to get the noise level even lower. Anyway, with one slightly sentient enchilada in hand, we could take some proper readings.
Is that special sentient sandwich?
We started with the array that was grown in neurobasal media. Under the microscope, they looked like nice neurons, and we saw them fire nicely before. With the machine, once again, we see nice big spikes, and what is immediately obvious is that some electrodes are picking up much more signal than others. When we inspect the array under the microscope, we find that those electrodes showing a strong signal had a neuron parked with its butt directly on top. I can see why the instructions say to try and get as many neurons as you can into the center of the array, because the ones sitting on the pads are picked up much more clearly. Now, with that baseline set, let's have a look at how the DMEM array looks. Once again, there are still lots of spikes, but their magnitude is dramatically smaller than the neurobasal. So perhaps this is the difference that Dr. Potter was talking about. Maybe this is more like what you get in an actual piece of brain tissue. But until we test the system with a slice of brain, we're not going to know that for sure. When we look at these cultures under the microscope, though, they definitely look odd. The cell morphology is different, and the cells look much more like fibroblasts than neurons. But that could just be a quirk of them growing in this media. However, just like with the electrical results, with the dye, seeing any kind of twinkle of activity was really hard. So it's actually hard to say at this point which is better. Are the big spikes in morphology that we saw with neurobasal an artifact of the growth media? Or is that what happy neurons are supposed to look like and behave like, and the DMEM ones are the ones that are underperforming? Let us know what you think below, especially if this is your field. Alright, we've now covered everything other than our homemade arrays, so we can finally talk about them. And then, afterwards, we'll play with the stimulator functions of the machine and see how the neurons respond. So, how did we make our new neuron arrays, and what's different this time? Well, as a quick reminder, in the last video we started exploring having our arrays made out of PET by a normal PCB manufacturer. But one issue we ran into was that there was a limit to how small of a hole they could cut in the coverlay. These PET circuits are actually 2 layers of plastic sandwiched together around a delicious metal middle. And to expose the metal for the electrodes, you need to physically make a hole in the top plastic before it's glued down in the final manufacturing step. Last time, this meant that instead of lots of little holes like we ordered, the company just cut one big hole around all of our electrodes, rendering them all useless. As we talked about last time, if your electrodes are too big, it actually makes your signal weaker, because you're averaging out the activity of many neurons over a large area. To get around that issue, this time we tried to be clever. We ordered small, thin holes and had the electrodes just barely poking out from underneath the plastic, so we could get our tiny electrodes while still working within the constraints of the manufacturer. And the big empty spaces leave a flat surface for the neurons to grow on and be observed through. We made and ordered 3 different versions, each designed to test different ideas on how to implement a game-playing array. The first uses square-shaped holes with multiple electrodes poking into each. The idea is that these simulate what we're calling action groups, or collections of electrodes all associated with one particular action that the array could take while playing a game. We tried different sizes and spacing to see how that affects things. The second test uses arc-shaped cuts, and we tried different spacings and radii to see how those affect the signals. The thinking with these is that these could be different inputs to the array, like the positions of different targets, walls, or items in the game world, though these pads could just as easily be set up as action groups the same way. And finally, there is this one. Unlike the other 2, this one is specifically designed to play a specific game. While we were thinking about how to make an array play Doom, we figured that it might be good to start with a game that has as few choices for the array to make as possible. Lots of data input for it to process, but only a small number of actions it can take. That way we can build up to the more complex case of Doom. When we were thinking about a good test case, what came to mind was an even older game called Asteroids. In Asteroids, you control a ship and have to shoot at the asteroids that are flying around, while trying not to get hit. But if we were to pin the ship to the middle and prevent it from leaving that spot, it becomes more like a tower defense game. So, in theory, this should be simple enough for an array to contend with right off the bat. All it's got to do is turn and shoot, so just 3 actions, left, right, and fire. But we can put a ton of targeting data in by giving it the azimuth and distance of the targets. Though, while we designed this array with this special version of Asteroids in mind, we realized that the same control scheme could be used to play Space Invaders just the same. Left, right, and shoot are your only options, and there's lots of targets to shoot at. Now, before you get your hopes up, no, we aren't going to test that today. While this system is amazing, it's designed for researchers, not a bunch of crazy nerds. So it's got an amazing recording and stimulation system, but it can only make 2 stimulation pulses at a time. Whereas for playing a game, we actually mostly need stimulation electrodes and only a very small number of recording electrodes comparatively. That does mean we need to build our own system, but that's okay. We've already got it designed and we think we know what parts we need. It's going to be built around the amplifier and stimulator chips from Intan Technologies. They're purpose-built for working with neurons in the sort of ways we have in mind. The one downside is the $10,000 price tag for all the bits we need. For today, we're just going to use the built-in stimulators of the current system and see how our arrays handle stimulation if the neurons grew properly in them. The PET arrays that we ordered are great, but they're super flexible and bendy, which is not helpful when we want them to be a solid array that we can handle. So they were mounted onto a glass plate backing using UV resin. Then to make these into actual culture wells, we used marine-grade silicone and some sections of polycarbonate tubing. Since none of this can be autoclaved, the plan is to sterilize everything chemically with alcohol, and all of these materials are inert to that. Then, just like the commercially made arrays, we turned some Teflon retainer rings on the lathe. Though we did choose a larger O-ring. Not because we thought it would be better, but because I'm an idiot and forgot to press order the day before the experiment when I was supposed to buy them. So we used what we had on hand. Whoops. That said, when the new O-rings that were the right size arrived, we did a little Indiana Jones switchy, but in the end, the clamshell and the head stage still couldn't fit over the O-rings or lids. So we moved the head stage into the flow hood and incredibly carefully removed the lid, closed the clamshell, and then fit the lid back on through the hole in the head stage. And we did the same for the reverse later when we needed to get it back out. Not ideal. So next time we're going to either go with a different mounting option or to make the wells less tall and thinner. One last note is that since DMEM was suggested to be the best, that's what we used to grow the neurons in our homemade arrays, so their activity was similar to the other DMEM test. There were still lots of clear spikes, but none of them were as big and punchy as we got with neurobasal media. Finally, let's play with the stimulator functions. The software lets you define an arbitrary waveform, so you can stimulate the neurons however you want, and you can choose which electrodes to stimulate. We were mostly interested in how the signals propagate and how the neurons respond, so we did try a variety of different pulses and electrode arrangements. What we found was that, as you'd expect, the neurons nearest to the pulse tend to fire really hard. But what was interesting was the increase in activity after a pulse, even far away from the stimulated electrode. And we saw the same behavior on all the arrays, including the homemade ones. These are pretty crude tests, but this video was already getting really long. Next time we're going to go through much more thorough experiments to see what exact stimulus patterns we should be using, and how they affect the dynamics of the neuron's activity. But importantly, our arrays seemed like they worked about as well as the commercial ones, which is a huge improvement, and means that next time we can focus on tuning their exact design and getting the neurons to actually play a game. And while of course Doom is the goal, we might only manage asteroids in the next video. But even that would be a huge accomplishment. No matter what, we are tantalizingly close to getting neurons to play a game. To commemorate this momentous occasion, we're doing something very special. If you've been following along with this series and ever thought that you'd like to have one of our arrays for yourself, now you can get one. And best of all, it's mounted to what might just be the coolest poster we've ever made. Not only is it screen printed by hand here in Quebec, we built secrets upon secrets into it. In white light, you get to enjoy this awesome design, but turn out the lights and the magic begins. In the dark, the bright phosphorescent layer lights up to reveal extra details and the glow of stained neurons. But if you use the UV light that comes with the poster, you can reveal one more secret in the form of a hidden message. If you want one of these amazing posters, then head to the link in the description or just head to thethought-emporium.ca and check out the other amazing items we have in stock. Between patrons and posters and merch sales, your support goes such a long way in helping us keep this project moving, so thank you so much in advance. We're going to be making as many of these posters as we get orders for, but they are made by hand, so it's going to take a few weeks before the first batch ships out. As an added bonus, the first 50 people to use the code TORMENTNEXUS will get 5% off their order. So be sure to check out the link below and get your poster today. And for those interested, there will be a ton of links to papers and other stuff in the description. That's all for now, and we'll see you next time.